Thank you very much. At the outset, I would like to thank the organizers for this opportunity. In fact, to be very frank, I was a little surprised when asked. I'm a rather modestly accomplished researcher, and it's especially daunting to try and offer advice to this super talented group of young scientists who are at the brink of starting their own careers. But over further interactions, I realized the goal was actually rather more interesting. It was to present a diversity of career trajectories to the audience and perhaps showcase the range of strategies that has been adopted by Indian scientists with various levels of success. Of course, this diversity of strategies is not unique to India, but talking about a specific context will probably help me connect with you, most of whom are independent, starting independent academic positions and are justifiably concerned about what lies ahead. So science is global. As scientists, we hopefully try to uncover universal mechanisms of how the world works. However, the actual practice of science is rather different, and it is contingent on the local conditions. Things like where you are situated, who do you talk to, who do you work with, what kinds of funding is available to you, etc. So the practice of science is rather contextual, and it is the tension between the global demands of science and the local circumstances that ends up shaping the exact nature of the questions we ask. Also shaping how we ask these questions, also shaping how we attempt to answer these questions. So what I will say in the following slides is going to be my individual story. So it's not going to be prescriptive. It is not necessarily going to give you universal solutions. But our hope is that it will alert you to be aware of specific local conditions and when necessary, be able to leverage this in the service of the science you want to practice. So let's start with this. Now, invitation to give a mentor talk at a young investigator meeting is also a rude awakening to the fact that you're no longer a young investigator. And which is probably fair. If you spent about 10 years or more in an independent position, one needs to move on. Cycle of life, etc., etc. This animal skull shown here actually literally dates me, not very prehistoric. But this animal skull was actually found in the land where IISER Pune campus stands currently. At that time when I joined, there was no campus. And some adventurous students, I have no idea what they were doing in this desolate field with a few sparse trees, managed to find this and brought it to the lab. Maybe I should start by sharing what my interests are and how they evolved. As Vasudha mentioned in her extremely generous introduction, I started off during my PhD being interested in precision of signal transduction systems in generating very precise outputs. And also state differences between signaling in different cell types and even pathologies. The interest was how are these precise outputs generated? Subsequently, this sort of work got me interested in this other major system that is able to generate extremely precise outputs, which is the nervous system. And the nervous system is able to do this through a combination of both hardwired circuitry, but also functional plasticity. And this now continues to be my interest in once I started my lab in IISER Pune. The questions we have tried to address and are the main interest in our group in IISER Pune are of two classes, not necessarily directly interconnected. The first is looking at stereotyped and long-term hardwired connectivity. And one part of this work, as I will show you, focuses on axon guidance. I had worked on axon guidance as a postdoctoral fellow, but then we were more interested in looking at these chemotactic cues that position neurons and their processes in the neural circuitry, how they are presented, and what kind of cellular signaling they might be inducing. In my own lab, I decided to make a shift and focus on the remodeling of the cytoskeleton within the neuron, which actually allows the physical process of movement of neurons and their processes to happen. The second thing we got interested in, related to long-term connectivity of neurons, is homeostasis of neurons, especially looking at how they generate physical forces and how physical forces impinge on the neurons and what consequences they might have. The second goal in the lab has been looking at plasticity in neural circuits, plasticity in the same circuit to generate a diversity of behaviors. And here we have focused on neuropeptides. So this is how we started. When I joined IISER, we didn't have a campus like we have now. We were located in a temporary space in a commercial building above a local bank known as the Cosmos Bank. The building was not known as IISER Pune building, rather as the Cosmos Bank building. And when I joined, there were 3 people who had joined before me, but these premises were entirely empty. We had managed to put in benches, etc., and that was it. And of course, the first goal was to try and fill this up. And we navigated a lot of things. We navigated the process of figuring out how to order things. These are the first set of chemicals which came for the first 3 labs that joined IISER. And we also navigated to recruit people into the lab. And it turns out also to the family by other means of biological experimentation. And finally, the lab got started. Now, here is a little aside. We had a child soon after joining IISER Pune, and that was a major challenge on the time I could give to the lab. But that had to be navigated. And I would also like to point out that though I talk of this challenge while having children and starting up a new lab, within families, even within families which, despite awareness and willingness to support, the burden of childcare, and this is not limited to just infancy of the child, but continues to go on. So this burden is disproportionately borne by the woman. And this is important in my trajectory because my wife also joined IISER Pune at the same time to start her own group. The burden which I'm saying is borne disproportionately by women, even if families are aware and supportive, is not merely physical, but it's also a significant cognitive burden. And I hope meetings like this and beyond will continue to discuss these issues and in order to try and develop equitable solutions and support systems. Anyway, so that was some of the human and infrastructural aspects of the lab. But I want to start a neuroscience lab, and I don't really trust neuronal cell lines to represent neurons accurately, and certainly they don't behave, at least in the context I'm interested in. So we needed model organisms. And remember what I showed you of our lab. This is in a commercial building with no hope of an animal house being set up for at least several years. So what I figured out was try and train myself and staff in the lab to use 2 systems which we had never used before. And this was the developing chick embryo, and the second was the zebrafish system. And there were local advantages trying to set this up. Venky's chicken, which many of you might be aware, the Venky's farm and their various associated institutions are located close to Pune. And this gave us a source of fertilized eggs fresh from a poultry where we could get them in a timed manner in order to do developmental experiments. Of course, I knew that this would be a good source of neurons, and looking at early development of the neural system, this couldn't be extended to look at behavior, which was the other interest in the lab. And hence we had to develop the second system of zebrafish, which was done in a modified kitchen in this building here, and starting off with a few aquaria, which we set up from a local fish shop, aquarium shop. Finally, of course, this has now extended into automated aquarium systems, and now we do CRISPR mutants, we develop transgenics, etc. But it had very humble beginnings. The second thing we needed to do for the kind of research that I was heading out to start, especially involving the cytoskeleton, is to have support for microscopy. And over the years, starting right from the beginning with the help of the administration, and what we managed to do was set up a very nice facility. Now, before I move to that, I want to point out a lot of these solutions, including the choice of model systems, were due to very specific local contexts. The microscopy facility that we set up in that temporary building has now expanded to about 4,500 square feet facility in our current campus, which now houses a large number of modalities of imaging. And however, I want to stress here, though this is actually a very local solution, which became very important. This is because we've realized over time that expensive infrastructure is best managed and sustained over a period of time in the form of facilities rather than equipment that belong to a specific lab. This sort of organization also buffers maintenance of these systems against the vagaries of grant cycles. Facilities do few other things that individual equipment may not be necessarily efficient at doing, which is they actually support a broad range of training options where people can get trained on modalities that are not necessarily being used at that point by your lab, but may become useful if they are available locally. Also, this holds true for development of new technologies locally, which can become useful at a later point of time. So once we had model systems to work on, we had a semblance of microscopy. Of course, the bigger picture here is one of our current facility. It started off on a much smaller scale. We were actually able to do things that we wanted to do, including image neurons growing live in a zebrafish embryo, and also do cell biological imaging of neurons isolated from chick embryos. So what my lab did was to try and identify novel cytoskeletal regulators in neurons. And we did identify a few of these using unbiased approaches. And we have been studying some of these. A small point I would like to make here is some of the regulators that we actually studied in significant detail at the time of our identifying them was not associated necessarily with anything known, especially any known human disorders. A couple of them, while we have been working on them, have actually become associated very closely with cognitive impairment and other neurodevelopmental disorders. So this may be a little plug for chasing things that, at that point might not necessarily have a direct influence on some health condition, but often by serendipity turn out to be fairly important. So what kind of things do? We look at dynamics of cytoskeleton in cells. Unfortunately, my movies aren't playing, it looks like now. We look at physical cell biological properties of cells, for example, measuring traction forces generated by neuronal growth cones. We look at what happens when these proteins are disrupted in developing embryos. Do they change neuronal trajectories? So what happens at the level of neurodevelopment in an organism? We also look at behavior. This movie would have looked nice if it played, but basically tracking behavior of zebrafish using code and behavioral systems that have been developed in-house. And sometimes when necessary, even look at single molecule dynamics, in this case of microtubules and actin filaments. With this range of investigative approaches that we were able to set up, we've managed to identify a lot of novel functions for a couple of novel cytoskeletal regulators. An example of this would be this protein formin 2, which we found to be involved in neuronal outgrowth and guidance. Despite being formin, which does some special regulation of the actin cytoskeleton, I'm not going to go into technical details in this talk. We were actually able to show that it has additional ancillary activities which actually seem to dominate in the neuron. This involves coupling the cytoskeleton to the adhesions that neurons make as they move. This protein we were also able to show is one of the few proteins that physically couple the actin and microtubule cytoskeletons to coordinately regulate them. And we found that they maintain other unusual actin structures which are required for branching, and later have shown in animal models that this protein is required for development of circuits, and lack of this protein results in maldevelopment and defects in behavioral outputs. The other thing we were looking at, so this is the neurons that we are talking about, and this flat structure here is the growth cone. But no, all of our work had focused on the growth cone. What happens into the growth cone as it moves? How does it generate forces? What happens between the actin and microtubule cytoskeletons? But what we also realized while doing this, that while looking at the growth cone, we were ignoring the axon behind it. As the growth cone moves, is the axon passive? Is it simply like a tail that the growth cone drags behind it, or is it actually active? We also noticed that not only they were likely to be active, so this is how the growth cone is moving, you can see the axon, but you see once the growth cone is anchored between two stable points, here generated by using microcontact printing, the axon seems to develop a force which then allows it to contract, and you could see the cell body which was here now being dragged to a completely different position. So we now wanted to study the biophysics of these axons. How do they generate forces? What kind of forces do they generate? How do they respond to applied force? And we wanted to do this in a rather quantitative manner. And fortuitously, I encountered this gentleman, Pramod Pularkat, who's a biophysicist at the Raman Research Institute in Bangalore, and encountered him completely out of serendipity at one of the earlier IIM meetings. So here's a plug for the IIMs. At least in my case, it has actually generated a long-lasting and very useful collaboration. And here's a plug for another local interaction that has allowed me to ask questions that would not have been possible. So Pramod introduced to our work concepts from the field of materials and soft matter. Soft matter, remember before this, was to me something that had to do with toothpaste rather than neurons. But it turns out one can apply principles of soft matter and understand neuronal function in a way that wouldn't have been possible if I had limited myself to only traditional cell biology. With Pramod, we were able to actually show a couple of interesting things. We were able to show that the axonal cytoskeleton is actively contractile, not a passive viscoelastic process, but there are actomyosin processes, active processes that actually regulate the contractility. And more recently, we've shown a very unusual phenomena in axons. Most cells, when you apply stress to them, the cells, because of the underlying cytoskeleton, show a stiffening response. They become more stiffer as you apply stresses. But axons show a reversible strain softening. They seem to respond by becoming more soft as you apply more pressure. And what we were able to show that this is because of a periodically arranged spectrin, which a sort of organization which was only discovered 7 years ago upon the popularization of super resolution microscopy. Though the functions of this are only emerging, we were able to show that this periodically arranged spectrin actually act as molecular shock absorbers to buffer stresses on neurons, on axons. And this is particularly important if you think of the organization of the axon. These can be very long, depending on how tall you are. It could be over a meter. If you are a blue whale, it could be 30 meters. And they are very thin, half a micron in diameter. And they are often subjected to a lot of stresses. A lot of stresses during limb movement. It's been recorded regular limb movement can result in 20% stretch. Also during normal activities like jumping, there's a shear stress about 5-6% on the neurons of the brain. And without a shock absorbing system, one expects that these neurites should have snapped. So this was an interesting discovery, entirely possible because I managed to meet Pramod at a local meeting and managed to sustain this collaboration. Other things we discovered that may help some of you to think about is undergraduate students. Remember, I'm at IISER, so we have a lot of BSMS students, all of them required to do a final one-year project in labs. BSMS students not only are extremely bright, their training is different from the traditional training of my generation, which is to say that they not only learn biology, but they also are very comfortable with quantitative methods. And therefore, they are also very useful to try out risky novel ideas, which you may not want to give straight away to a PhD student whose success depends on successfully executing these ideas. The other thing we learned that in the Indian funding system, if one writes a good, interesting proposal, one can still get substantially funded for this proposal with very little preliminary data. So this accidental meeting with Pramod at YIM actually resulted in not only some interesting stuff that I described, but in fact, more recently has led us to get a joint grant, which is a rather lucrative grant, which will allow us to apply some of these on human pathologies and see if they make a difference there. So this was another local solution that we have implemented, use of undergrad students efficiently and getting funding with relatively little preliminary data, which may not be possible elsewhere in the West.
Aurnab?
Yes.
Maybe in a couple of minutes,
yes,
we have some interesting questions to discuss.
Absolutely.
Yeah.
The second part of our work involves neuropeptides. I was interested in motivational states and crosstalk with emotions and reinforcement. Here I met Professor Subedar in IISER Pune. I had no training in neuroanatomy. He's an expert neuroanatomy, so I can't distinguish my inferior olive from the bed nucleus of the stria terminalis, for example. He helped me learn all of this, and again, a local solution that resulted in interesting behavioral setups, activity imaging, as you can see at the bottom, with both fish and rodents. And we were able to come up with interesting behaviors and explanations underlying this at the circuit level that involved, for example, changes in olfactory sensitivity, hunger and satiety states inducing bistable activity states via neuropeptides, and also their role in modulating fear and risk-taking in rodents. So now you see a new model organism, and that was only possible because much later, with the support of DBT, and we were able to set up a facility which now allows us to do a lot of rodent work. And this again has all the advantages of this being set up as a facility rather than something for one single lab. Finally, the other big thing with IISER is teaching. Teaching has been extremely useful. This allowed me at least to structure and organize concepts. And this also, among other things, if you're a reasonable teacher, it's a good lure to attract talented human resource. Finally, what about giving back locally? A lot of this was being taken. I think this was dramatically shown by the local response to the pandemic, where IISER managed to set up a testing center entirely run by volunteers, and also epidemiological studies in the city of IISER Pune, with which I was also involved. I think my message is this: the global and the local are deeply interconnected. One needs to be aware of the local conditions and influences, because this enables science doing and one should think of actively enabling or leveraging these local conditions towards your science. I would end by thanking people in the lab, collaborators and mentors, the golden sunset that is always there at IISER, and people who have funded us over time. Thank you, and I'll take questions.
Thank you, Aurnab, so much for that stimulating talk. It's really amazing how you have literally localized your research. Your talk has given us the confidence that the solutions are really around us. We must look out.
Thank you.
Yeah, so there are a couple of questions, in fact, a few more. So Manasi Mishra asks you, how to build your network of mentors and peers when you have had some major shift in your location? I think that's a very interesting question out there.
I think one of the nice things that's happened for a little over a decade now is having structured networking opportunities, IIM being one of them. IIM has also expanded to local IIMs, which happen in certain areas of the country, like let's say a Western IIM or an Eastern IIM, etc. I think one needs to participate in these closely. These are often the initial networks that we start up with. The other thing that really helped us, and this I will credit to a senior mentor of IIMS and a part of the India Bioscience Board, is Professor Shashidhara. He encouraged all of us when we joined, he was the chair of the department when I joined, to actively meet anyone, any scientist who was visiting Pune city from anywhere in the world. And some of them were senior scientists, some of them were junior scientists, it didn't matter. We would meet them rather indiscriminately. And many of these have lasted in at least networks, which I still use. And there's also a reciprocity in building networks. Many of us, again, I know some of them are actually here, we have formed, sort of spontaneously a network have emerged that we often will even help looking at people's manuscripts. If it's an area which is closer to me and this new person is venturing there, this also, if nothing, creates a lot of goodwill. So I think interactions are important. Structured opportunities are now available and they should be utilized to the hilt.
All right, that's really, I think, insightful input for people who are starting up. I think a lot of us had these questions and these platforms can be utilized to tap that ideas.
Exactly.
So Priyankar Day asks a question, as a new faculty in IISER, how did in-house collaborations facilitated your transition as a young researcher? So in other words, how should a young researcher leverage in-house collaborations?
Yeah.
So internal collaborations specifically.
Yeah. So remember, especially internal in-house collaborations come in added flavors. So when we think of collaborations, we are often thinking of one scientific project that leads to an outcome where two or more groups have collaborated. But there's also this other thing where there is a local repository of knowledge which you can tap into, even a local repository of consumables, techniques, analytical techniques. So this helps a lot. What we did fairly early on is COVID shut everything down. But every other, we have a Wednesday faculty lunch meeting, over which, over lunch, faculty very informally present data to others. And this has actually led to a lot of local collaborations. Not many of these are necessarily projects, but actually supporting each other in ways of thinking or with expertise or with tools. But there have also been a number of actual collaboration resulting in scientific projects and papers. For me, the best of these have come with Professor N.K. Subedar when he was here as a regular faculty. This whole idea of looking into behavioral plasticity resulting from network dynamics, I couldn't have executed without his presence here and the support he gave in terms of teaching us neuroanatomy and peptides.
So, yeah, that I think people have seen the outcomes in that way, how locally faculty inside the IISER Pune system have collaborated. So there's another question, interestingly, like how tricky is it for young professors to reach out for collaborations with established faculty? Yeah, I mean, how do you reach to senior professors, essentially?
You know, you have to bait them with the science. That's what really gets most of us going, senior, junior, in between, right? I think if you have interesting science and where they can genuinely leverage, your expertise as well. So scenarios where it's not one way, where you actually are able to engage people, by also adding value yourself, are the ones that work best. There's also be aware that especially setting up interdisciplinary collaborations like the one I have with Pramod for some time now, requires all of us to adapt sometimes, especially in the language. The language of discussing the same cell biological phenomena to a physicist, by a physicist, and the way a cell biologist would describe are sometimes at odds, and one needs to make efforts to bridge this. This is not insurmountable at all, but the effort needs to be there. So my suggestion is the bait is always good, interesting science.
So there's someone who wants to ask a question. Can you please, Nishant?
So Nisit has permission to unmute and ask the question. Nisit, go ahead.
Yeah, thanks. Hi, Aurnab. Thanks for the wonderful talk and showing like real difficulties in setting up the lab. And so my question is that since you work in the field of cell mechanics, which is actually a niche field in India, where, as you just said, that you have to talk to biophysicists, how difficult it is to, get funding for something like this. So that's my first question. The other question which I have is, what would be your suggestion for developing collaboration for someone who is new, who doesn't have too many contacts in India, and who wants to come back to India? You see, because it would not be possible to attend like local YIMs in that case. So your opinion and advice would be really appreciated.
So I'll take the second one first. I was in a very similar scenario. Any of us who've not done their PhDs in India actually do not know that many people here necessarily, when you come in at least. I frankly did not necessarily set up a lot of collaborations before I joined here, but it has happened since. It has happened with me. I have had people approaching me who are looking to come back to India or have got a job but not yet moved to the country physically, back to India physically. Initiate conversations and actually start off things. In fact, with one of them, once he moved back to India, we are already sharing reagents and doing things together. Frankly, you have to go out and approach these people. And as I said in the previous question, that, interesting science always, will get, people interested and they'll be ready to work with you. So however, if you ask for things that are simply procedural and not that interesting, your response rate may not be as great. But there have been people even when I came back who have helped me even with very simple things. In fact, knowing that I was joining IISER Pune where labs weren't set up at that point, there were a couple of neuroscientists elsewhere, not even in the city, who in fact offered me to come and use their bench space for a few months if I needed. And that was just remarkable. Absolutely didn't expect it.
So although there are, several questions, we'll take one last question.
Sure.
Can you elaborate on the two-body problem? That's a very interesting question because you prompted about that.
The physics two-body problem or the in two-body?
The biology human two-body problem.
Yeah. Again, I don't have a solution. I will tell you what we did, right? And remember, this is a little over 10 years ago. Things were different. Many of us at that time would actually end up getting more than one jobs, especially because IISERs were coming up and the established institutes were already there. So what we did so I know one strategy is to stagger the application. And it depends on if the two individuals are not ready at the same time, right? But if I know the strategy is also to stagger even when both are equally ready. And the idea is one person gets it, accepts the job, and the other person tries to find a job in the same city. Which of course means that you're now restricted to only a few cities which have a diversity of labs and institutes which will take both of you, will be able to absorb both of you. By the way, before anything else, I should say, there are places where spouses are not hired. I do not know if these are written rules, but they are certainly practiced. But strikingly, the newer institutions that have come up in the last decade or a little bit more have actually, it seems, proactively not necessarily adhered to this. Many of us in IISER Pune, and I know in other IISERs, for example, are two people sometimes in the same department, sometimes in different departments. So what we did, in short, was we applied together. Wherever I applied, I indicated, no pressure, that my spouse, such and such person, is also applying. If you can coordinate our applications, this would help us tremendously. And she did the same. And surprisingly, again, things were a little different then. We had options in more than one city where we could, both of us would have had jobs. And then you take into account my favorite phrase of the day, local context, and choose what is ideally suited. But again, I don't think there is a very prescriptive answer I can give for this. The recruitment policies and what areas being recruited vary, which one doesn't necessarily know from the outside unless you're already in the institute. But I think a good strategy is to be right at the beginning, state that this would be an ideal scenario. One is considering moving with his or her spouse, at least to the same city.